Introduce today's speaker, Rebecca Degnan, a PhD student at the University of Queensland in Brisbane. Rebecca, I'm just passing the screen to you if you'd like to accept the invitation to present there. Now, before starting her PhD, Rebecca completed an honours degree in molecular cell biology. In addition to studying, she has been working with the Department of Agriculture and Fisheries for several years under the supervision of Louise Shuey. Louise will be familiar to some of you from collaborations with New Zealand researchers. And I understand Rebecca and Louise have been making great progress with RNAi. We should be ready to hear about that now, but unfortunately I can't see your slides there, Rebecca.
Oh,
now here we go.
really?
Now I can. I'm sorry, the gremlins have really got me this morning.
You've got them?
I think that we're all good to go if you'd like to begin your presentation.
Awesome. Thanks, Renee, for the introduction, and I hope everyone can see that okay. So it's wonderful to be here and have the chance to present some of our recent research with you. And I'll just begin by paying my respects to all First Nations elders and descendants. And I'd like to highlight also the significance and importance of Aboriginal and Torres Strait Islander knowledge, and I think it's important that we continue to acknowledge the contributions of Indigenous sciences and also the practices that have preserved this land for 70,000 plus years. So today I'm speaking to you from Brisbane or Meanjin, where we're very privileged to live and work on beautiful Yagra and Turrbal country. So I've probably met quite a few of you before, but for those of you who I haven't, my name is Beck. I'm in the first year of my PhD at UQ, and I've been working on Myrtle Rust for probably 3 or 4 years now. So I'm just going to share with you some of our kind of recent results in RNA interference mediated management of Myrtle Rust. And of course, these results are very much made possible by my supervisors and also my co-authors who are listed up here. So I won't spend too long on the specifics of Myrtle Rust because I imagine I'm probably speaking to a pretty well-versed audience. But I'll just go over the basics kind of just in case. So Austropuccinia is the causal agent of Myrtle Rust. It infects plants of the family Myrtaceae. There are over 2,000 species of Myrtaceae native to Australia. It infects new growth, and symptoms range in mortality from leaf spots and blight to whole tree malformation and mortality. We now have also over 50 species of Myrtaceae listed as conservation priorities as a result of Myrtle Rust related population decline. And many of these hosts are also of high environmental, cultural and/or economic importance. So just to kind of take a step back outwards for a moment, the rust fungi are actually the largest group of plant pathogens. You have these kinds of agricultural rust like soybean, coffee and wheat rust, and then kind of garden variety rust like geranium, monstera and frangipani rust. If you're in or around Brisbane, you've probably encountered frangipani rust plenty of times before. And then of course there are also the environmentally significant rusts like Myrtle Rust. So yeah, the rusts are really diverse and also really destructive. And I think this kind of highlights the full extent of possibilities for broad-scale RNA interference mediated management of not just rust fungi and not just Myrtle Rust, but also plant pathogens as a whole. So I've mentioned RNAi a couple times now. I'll just go through briefly what it is and how it works. So RNA interference, or RNAi as I refer to it, is a post-transcriptional gene silencing mechanism. It is largely conserved throughout eukaryotes. Like most things in biology, there are a few exceptions. But yeah, for our purpose, it's largely conserved. And it's also thought to have evolved as a defense mechanism against viruses and transposons. Transposons are those little genes that jump around your genome, wreaking havoc. And it's triggered by the recognition of these longer double-stranded RNAs, or dsRNAs. The dsRNAs are cleaved into small interfering RNAs, or siRNAs, by an RNase 3-like enzyme called dicer. The sense strand of those siRNAs is cleaved off, and the antisense strand complexes with a protein called argonaute to form the RNA-induced silencing complex, or RISC. It's a nice, easy name to remember. And then RISC then guides sequence-specific degradation of homologous mRNA, resulting in silencing of a target gene. So that RISC is going to degrade mRNA that has homology to that original double-stranded RNA. And in the case of some organisms, if they have these RNA-dependent RNA polymerases, or the RDRPs, you can also get amplification of that signal by RDRPs, kind of leading to a feedback loop and a continuous silencing effect. And the really cool thing about RNAi is that this is something that already exists in most eukaryotes. So we can kind of take advantage of it and manipulate it and use it as a means to protect plants against a variety of plant pathogens, including against rusts. So to walk you through how RNAi kind of could work in this system, in the Myrtaceae Myrtle Rust pathosystem, what we do is we choose gene targets in the rust itself, and you want to choose genes that either the rust can't live without or it can't infect without. And then we synthesize double-stranded RNA with homology to those genes and just apply that exogenously via spraying to the plant itself. So it's just a suspension of double-stranded RNA in water sprayed onto the plant. And there it's taken up and processed, resulting in degradation of mRNA, silencing of those essential genes, and ultimately a reduction in disease symptoms. And when I first drew this diagram, which was probably a couple years ago now, this is kind of what I thought was happening, or what we thought might be happening. Like some or most of the pathway was happening actually in the plant cell, and then at some point maybe the siRNAs were being trafficked into the fungi, and then the mRNA silencing was happening there. And now, based on our results, we think it's probably more likely that most of the pathway actually is happening or can happen inside in the rust itself. So it probably looks a little bit more like this. We spray the double-stranded RNA onto the plant, and then from the leaf surface, it's taken up directly into the spore, and that whole pathway, right from double-stranded RNA down to mRNA degradation, is happening in the rust. And for that to be the case, that would require Austropuccinia to actually possess those RNAi machinery genes, so the RDRPs, the dicers, and the argonautes. So that was kind of the first thing to do to, check if this machinery actually was present. So this is a phylogenetic analysis, a really awesome, comprehensive analysis done by one of my supervisors, Alistair McTaggart. And he found that these RNAi pathway genes, so again, the RDRPs, dicers, and argonauts, were conserved across 8 families of rusts, including in Austropuccinia. And then we also know from the literature that exogenous RNAi has been shown, also shown experimentally in a 9th family, and this is the family that soybean rust is a part of. So we know that the machinery is present and it's there, and the next thing to do is to kind of work out if it's actually functional. So first we needed to know if the spores of the rust could actually take up double-stranded RNA. So this was some microscopy done by another of my supervisors, Anne Sawyer, and she used Cy3 labeled double-stranded RNA. So that's just double-stranded RNA with a little fluorescent tag on it that allows you to visualize it. And she looked at uptake in both Austropuccinia, spores, and spores, and also in Coliosporium plumeriae, which is the causal agent of frangipani rust, spores. And so you can see in panels A, B, and D, which are all of the spores, that double-stranded RNA is taken up very readily from the environment. Interestingly, it's not taken up into spores, and we don't really know why yet. It's a bit of a question for the future. But importantly for us, it is taken up very efficiently into the germinating spores. She also found that the double-stranded RNA is taken up very early in germination, almost immediately. So you can see in panel A where there are these germ pores, not even germ tubes yet, but just like the very beginning of a germ pore. And the spore is already fluorescing with that Cy3 labeled double-stranded RNA. So we know that the genes are there, we know that the spores can take up the RNA. And the next thing to do was kind of to test it out and see if it was a possibility for this system. So like I briefly mentioned before, we wanted to choose genes that either the rust couldn't live without or it couldn't infect without. And so we chose quite a broad range of targets to screen. We chose 11 targets altogether with a range of functions across like housekeeping genes and also more general pathogenicity, and then also a few genes that were upregulated in the Historia, which is that fungal feeding structure that biotrophic fungi set up. And those are just all of our double-stranded RNAs on a gel. You can see that they're all, or mostly all around 300 to 400 base pairs in length. And we did test one that was a little bit bigger as well, just as a comparison. So we started with these detached leaf assays where we screened all 11 targets plus a non-specific GFP control. And we used Syzygium jambos detached leaves for this. So basically what we did is we just cut off the leaf at the petiole. In a petri dish, pipetted a little bit of agar and kind of a crescent moon shape down the bottom, and we'd stick the petiole into that. In each plate, we'd have a control leaf and a treatment leaf, which would be leaves of the same age, just to kind of minimize any biological differences. And then the control leaf would receive just the Austropuccinia spore inoculum, and the treatment leaf would receive that same inoculum, but with the addition of double-stranded RNA. And we would leave them for 2 weeks and then kind of observe the symptoms after that 2-week period. And this is what we saw. So this is an example of one of our treatments, double-stranded RNA3. You can see the control leaf where we've pipetted the Austropuccinia inoculum. It has that classic Myrtle Rust symptom developing, the yellow pustules. And then on the treatment leaf, which received the same inoculum pipetted in the same places, it's almost completely inhibited. There's just a couple tiny little pustules there, but it's looking pretty healthy. So that was a very exciting result. And I've also just included the GFP here to show you, GFP being green fluorescent protein, which is obviously not a gene in Myrtle Rust, in Austropuccinia. So that's just to show you that when the double-stranded RNA is not designed to be homologous to a target gene in the pathogen itself, it has no effect, which is really good news for any kind of concerns about potential off-target effects or things like that. And these are just the full results of that assay. So 8 out of our 11 treatments resulted in a significant reduction in pustule number. All of these ones in green here to the left of the dotted line, those all resulted in a significant reduction. And that's also represented in this bar graph here. So because we had such promising results with that detached leaf assay, we wanted to know what was happening, like how the infection was actually being suppressed. So to do that, we needed to look at the actual physiology of the rust spore in vitro. And this was really made possible by these awesome PDMS artificial leaves. I'll call them artificial leaves in this talk. And these were innovated and designed and developed by Grant Smith and his colleagues in New Zealand, Volker, Rebecca, Ashley, and Sarah, and very kindly provided to us. And these are really what made, I think, some of our coolest results possible. So we're very grateful for this contribution. And they've been so much fun to work with. And the setup of this assay is really very similar to the detached leaf assay. We basically just pipette the inoculum on top of the artificial leaves. But in the case of this assay, we don't have to wait 2 weeks, we only have to wait 24 hours. So we wanted to look at inhibition of germination, and then also what the impact on a few kind of crucial infection structures was. So we tested 3 of our best double-stranded RNA constructs in this assay, and you can see in this top left figure, in the case of all 3 treatments, there was a significant reduction in germination. And then the kind of interesting thing is that, so in some of our treatments, we did still see a bit of germination, but I guess if the spores are germinating and they're forming that germ tube, but they're not forming an aprosoria or an infection peg, that spore basically is not or can't result in an infection, because the infection peg is really what's needed to actually penetrate into the plant and then set up that hostorium. So we also quantified these other infection structures, and then you can see, even when we did see a bit of germination, if we look at aprosoria and infection peg formation, there's basically nothing going on. We saw maybe 1 or 2 aprosoria, but yeah, very, very little to report. And this is how the spores looked. So if you look at our 2 controls here, the negative control and the non-specific GFP control, you can see these really awesome aprosoria and infection pegs forming. And like I said, this alone was so much fun for us, just to be able to kind of look at that. We spent so much time sitting around looking at these photos and taking photos. And then if you compare it to the treatments, sometimes we just didn't see much germination at all. Other times we saw these really short, stumpy germ tubes. Sometimes the germ tubes were long and withered. Sometimes we saw a little bit of swelling at the end of a germ tube, but never those like really nice pronounced aprosoria or infection pegs like we were seeing in the controls. So this is really cool, and basically told us that Austropuccinia is able to actually take up and process the double-stranded RNA, ultimately resulting in an inhibition of those infection structures and also germination. So we also repeated this same experiment in Colliosporium, the causal agent French penny rust, and observed pretty much exactly the same results. So the aprosoria are a bit less nice in Colliosporium, they're a bit less pronounced, but you can still see them happening here, and you can kind of still see the pretty drastic difference between the treatments. And also when we quantified it, there's really not a lot in the way of germination or infection structures going on in any of the treatments. So once we had those in vitro assays kind of set up and running smoothly, we decided to have a look also at different doses or different concentrations of double-stranded RNA. When we started our experiments, we were using quite a high concentration, 100 nanograms per microliter, which is definitely the upper end of what's used in the literature. And we chose to use that upper end just because we didn't want to be limited by the concentration we were using. We wanted to kind of make sure that wasn't the problem if we were seeing any issues. But once we had it kind of all set up, we knocked it down a bit. We did a few dilutions to see what the kind of dose-dependent impacts were. And we did find that in the case of all of our treatments, and also in both French penny rust and Myrtle Rust, the higher concentrations were more effective at inhibiting germination. In some cases, we still saw a significant reduction in germination when we used 10 nanograms per microliter, but it was always better at 100 nanograms. So that was kind of just interesting to like corroborate what we had already thought might be the case. And yeah, it was nice to be able to see those results and see how kind of smooth that trend is. So we also wanted to look at this in the actual Myrtaceae Austropuccinia pathosystem and see how it actually manifests in planta. So we did a few different in planta assays. We started by mixing the double-stranded RNA with the Austropuccinia inoculum and co-inoculating all together. And when we did this, we just did 2 of our treatments because it does use a much larger quantity of double-stranded RNA. When we did this, we saw a significant reduction in diseased portion of new leaves. So we measured this as kind of, yeah, the disease coverage on potentially susceptible leaves. And in the case of one of our treatments of RNA3, we actually didn't see any rust at all in any of the plants, which was really awesome. So we then kind of wanted to play around with a few different time points, applying the RNA before and after infection to see what might happen there. So when we applied the double-stranded RNA at 6 days prior to infection, so as a kind of protective assay, we saw a significant protection with RNA3, but not RNA1. And this kind of follows the trend from the co-inoculation assay as well, so it makes sense. And the thing with double-stranded RNA, with naked double-stranded RNA, is that it's not that stable. So compared to single-stranded RNA, it's super stable, but it's still not that stable. It will only last on a plant kind of 5 to 7 days, depending on things like rainfall and UV and things like that. So 6 days is really kind of right at the end of that stability window, and probably by 6 days, a lot of it had degraded. So we know from that dose assay that lower amounts of double-stranded RNA are going to mean less effective inhibition of germination. So this is kind of to be expected, and you know, in the future we might need to try complexing the double-stranded RNA with a nanocarrier or something similar to improve the stability. So from those protective results, what I've been working on really recently is using double-stranded RNA to actually treat or cure myrtle rust after an infection has already happened. So these results are super new. I don't think we've really presented them before, so it's pretty exciting, and it's also still ongoing. I'm currently repeating this experiment and kind of getting some additional data and things like that. So what we did is we applied the double-stranded RNA at multiple time points post-infection. So we did 24 hours, 6 days, 8 days, and 14 days. And we basically chose these time points based on stages in the infection. So 24 hours is like the spores have germinated and are kind of, you know, starting to penetrate into the plant, but maybe still like sitting on the leaf surface a bit. 6 days is when you see the very first symptoms of myrtle rust infection in Syzygium. So you start to see that red pigment, like the red dotting on the leaf. 8 days is when you see the first pustules forming normally, and then 14 days is like an established infection. So this is the results from the 24-hour curative treatment and the 6-day curative treatment. And yeah, they're pretty exciting. So you can see in both cases, we saw a significant reduction in disease coverage in both of our treatments. And I've also got that in picture form for the more visual people. It was a pretty severe infection in the controls. Like, you can see that they're really getting pretty smashed by the rust. So the fact that our treatments were looking so good was really exciting. We did still sometimes see a little bit of rust, which I guess is to be expected, but overall they were looking pretty good. And I know it's a little bit small, but if you look on the bottom right-hand side here in double-stranded RNA1 treatment, you can see these kind of red pigments on the leaf. And that is what kind of starts to form at 6 days on Syzygium jambos. And what we would notice, because these plants were all scored and these photos were taken at 2 weeks post-infection. So what we found in plants that were treated with the RNA at 6 days post-infection is they would often have these little red dots on the leaves, but then the infection would never progress past that. They would just stay as those little red dots. So that's pretty cool and pretty interesting, and we kind of want to look more into the mechanisms of that as well, and kind of how or why that's actually happening. So in terms of rating the kind of outcomes of those later curative time points, the 8 days and the 14 days, it's a little bit harder to do because there's already rust there, there's already pustules, there's already an established infection, and it's not going to just disappear. So rating it as disease coverage didn't really work. So instead we waited until 6 weeks post-infection when the plants had time to actually start to regrow new leaves and things like that. And then we rated the plants on this kind of qualitative plant health/growth rating scale, with 0 being just totally dead and 5 being at least 3 new leaves per node. And so these are all of our control and treatment plants side by side, and you can see kind of straight away that the controls, they just don't look very happy. Some of them are completely dead, some of them had very severe defoliation, which is why you don't actually see much active rust, because most of the infected tissue had actually become so infected that it had just fallen off. And there's almost no new growth either. When there was new growth, the leaves were kind of very small and just overall not looking too great. And then in the treatments, of course, there's a bit of variability. Not all the plants were amazing, but overall, they look pretty good. And there's a lot of new growth. We saw almost no, if any, defoliation. And yeah, it's pretty kind of black and white from these photos, I think. And then when you look at how each of the treatments at each time point fared on our qualitative plant health scale, you can see kind of at all of the time points, the plant health or growth was significantly better in the treatments as compared to the controls. And like I said, there's a bit of variability. Not all the plants were amazing, but generally we saw much more growth, much more new growth emerging in the treated plants. And then I've also included here a kind of representative example of each of our controls and treatments for the 14-day curative plants. And again, these were taken at 6 weeks post-infection. So the kind of most notable thing is that in the controls, there's just a lot of defoliation, or the infected leaves are just totally dead. And then in the treatments, there's a lot of new growth, which is pretty exciting. So this is kind of the ongoing aspect and what I'm doing at the moment, and we'll probably be doing for the next kind of month or two. I'm doing this like a repeat of that curative experiment, but with a couple of additional steps. So I'm going to have those same 4 treatments, those same 4 curative treatments, and then also add a 5th one. But then the last 2 treatments are also going to become preventative treatments for when I'll then reinfect the plants and then run through this whole cycle again, maybe even 3 times, but I'm still kind of figuring it out. And I'll score disease symptoms and also plant health or growth throughout. So that would be really interesting to kind of see, how the plants fare at the end of all of that. And I guess ultimately the goal would be to kind of minimize how often we need to apply the treatment. So what would be really cool is if we could get it to like infecting and then 2 weeks later treating, 2 weeks later infecting, and kind of repeating that and seeing if the treated plants stay in better shape than the control plants. So the other really exciting thing that we're working on at the moment and are starting to kind of like really knuckle down on is this project investigating Austropuccinia effectors with our collaborators at Plant and Food Research in New Zealand, Grant and Rebecca. And in this project, we want to kind of more so use RNAi less as a control method and more of like as a tool to understand the role and function of predicted effectors. So in this collaboration, analyses of Austropuccinia infected leptospermum has revealed 3 candidate effectors or predicted effectors. So we're going to use exogenous RNAi to target and knock down those effectors both individually and also in unison to see if we can understand their function and maybe identify any redundancy or things like that. And yeah, I think it's a really cool opportunity to see what's going on with these effectors, but also how RNAi can be used as a tool in systems where we don't have available transformation procedures and things like that. So it's a kind of an exciting opportunity to work on a non-model system in a really innovative way. I think that is it from me. So yeah, thank you so much for your attention, and thank you to all of my supervisors and co-authors and all of our collaborators. I'm very lucky to work with such a kind of diverse and cool group of people across so many different institutes and organizations. So yeah, I think that's it. I'm very happy to answer any questions. My email's also there, so you can email me if you think of something later. And yeah, thank you again.
Great job, Rebecca. Excellent presentation, and great to hear you referencing how much fun you guys are having. Taking those beautiful images and seeing what's going on. So it seems that the RNA essentially induces the rust to commit suicide. At least
essentially, yeah.
That I interpreted your explanation at the beginning there.
Yeah, that was a pretty fair interpretation, yeah.
The plant may not even necessarily have a great deal to do with it.
Yeah, so we're still kind of trying to work out like maybe it can have something to do with it, but it definitely seems as though the rust can just do the whole thing itself.
One thing you mentioned, I was interested to get a bit more clarification. It's faster to work with the artificial leaves. Why is that?
So on the detached leaves, we have to wait for symptoms to develop, which generally takes about 2 weeks to see like the really good like yellow pustules forming. But on the artificial leaves, because we're not waiting for infection on a plant, we're just waiting for the spores to germinate and kind of begin those infection procedures. That happens in the first kind of 12 to 24 hours. They actually start germinating in the first like 4 hours, or sometimes even faster. And then you start to see, by 24 hours, you can see those infection structures like aprosoria and infection pegs. And because it's a biotrophic fungus too, you're not ever going to be able to get it to grow more than a day or 2 in vitro anyway. So yeah, it's a much faster assay, which is really cool.
Although I guess if you were looking to target genes that were expressed later, then you would be stuck back with the plant.
Yes, definitely. Yeah. So I think, for example, with the effectors, we might not see any impact on the artificial leaves. We might, we're not sure yet. It might require an interaction with the plant. yeah, but definitely some targets, I think timing is more important, but which with the kind of like essential housekeeping genes, really early on is fine.
Excellent. Well, I've got a few more questions, but we've also got some coming in from the audience, so I better stop hogging the question session and get started.
Sure.
So, of course, lots of feedback too on great talk and work and results. Did you test for off-target effects on related rust species?
Yeah, so the cool thing about RNAi is that it's very highly sequence specific, and because those small interfering RNAs are only like 21 to 24 nucleotides, there's a lot of like capacity for specificity there, but also we can screen all of our constructs or targets against a database that checks against kind of related organisms, but also potentially beneficial organisms that might exist in the surroundings. So you can kind of screen against beneficial organisms or related organisms and check for any potential off-target effects.
Sure, and I guess in the end, you either have the genes or you don't. And if you don't, then you shouldn't have a problem as a related species.
And of course, and all of our targets are designed like directly from the Austropuccinia genome as well.
Right, next question. I wonder if effectors needed for infection in Manuka are the same needed in other members in the Myrtle family.
Yeah, it's a very good question. Not one that I can offer much intel on, mostly because I don't know. I would think that they would be the same. But yeah, I guess we don't actually know until we test it. So that will be interesting to see. There's probably someone in the audience who knows a lot more about this than me.
Yes,
so
well,
yeah.
I should be careful because it's not my own work, so my understanding is not deep. But I know from work that Grant Smith has been doing that the rust runs the same kind of genetic programming, irrespective of what host it lands on, at least as far as he's been able to tell. So. And I believe that's one of the exciting things about what he's finding, that yes, it does mean that these kinds of approaches could be effective across all Myrtaceae species. At least that is my understanding. If Grant's here, perhaps he'll pop a comment in the question box or something like that. All right, a question that is going to be on the minds of many of us, of course, is around resistance, the big issue with more traditional fungicides. And I guess, the key again is you just target a gene that the fungus can't live without, and it's hard to imagine how resistance could occur. Have you got any additional comments to make on that?
Yeah, so I guess a few things. One, exactly like you said, if we're targeting, something so core and so essential, it would be very difficult for the pathogen to get around that. Kind of what we've discussed is that probably the only way for it to really actually evolve resistance would be if it lost its RNAi machinery. But RNAi machinery is actually important for a whole range of things, not just this kind of virus defense pathway. It's actually got a whole range of functions. So, and because it is so highly conserved, it's so unlikely that machinery would be lost. If we were targeting a specific gene and then that gene mutated, so it became resistant in that way, the cool thing about exogenous RNAi is that it's relatively easy to just choose a new target and design new double-stranded RNA and synthesize new double-stranded RNA, or just modify our target. That's kind of the difference between this and a lot of other more traditional control methods, is kind of the speed at which it can be developed. It really can be quite fast with the right kind of resources behind it.
And I wonder as well about the potential for targeting multiple genes at the same time. You've shown that multiple genes are effective. Is there an intention to look at whether you might target multiple genes at once?
Yeah, so you definitely could, and I think other people in other systems have. I guess it's kind of like the gene pyramiding thing. And I guess we haven't found that it's necessary yet, but it is definitely a possibility that yeah, you could look into. It should be possible.
All right, we have one more question here. This looks very promising. Indeed, it does. And of course, people are going to be interested to hear about moving forward. Are you working on a formulation of the RNA for field use? I mean, you mentioned you're only in the first year of your PhD. So I guess you could still go in a number of different directions. And also mention you might like to start using a fungicide in your experiments as a positive control for comparison. For example, triadimenol, which is highly effective against Myrtle Rust. So perhaps, yeah, you could talk a little bit more about your plans or perhaps your group's plans to move more towards application of these advances.
Yeah, so I think that is something that will happen in the future, whether it's something that will be done by me or by someone else, I'm not sure, because like you said, there are so many directions, and I guess in a kind of selfish sense, I'm really interested in kind of understanding more about like the core biology of rust and Myrtle Rust, and using RNAi as more of like a tool, like a molecular tool in these systems where we don't have access to things like transformation procedures. So that's kind of like my real interest for my my PhD, but of course at the same time, what kind of got me into this in the first place was like myrtaceae conservation and you know, finding a treatment for Myrtle Rust. So yeah, short answer, that will be happening. That is the direction that it's going in. But I don't know if that particular aspect, as in like the field trials and things like that, will be done by me.
Very good, and we have heard here from Grant, and I did make something of a misinterpretation with regards to his results, but we can clear that up now. And Grant saying what I hoped I said, yes, you did, Grant, was we see not changes in transcript profiles on the different Manuka resistance phenotypes, because he has really been focusing on Manuka down here in New Zealand. So it looks like Austropuccinia psidii is not responding to plant signals, at least early in the infection process. Don't have any data on other species, we'd need to phenotype first. Well, sounds like a good project for maybe another student.
There's lots to do, like you said.
Yeah, there certainly is. All right, well, that was excellent. We're going to wrap it up there. And thank you once again for your presentation, which will be available as a video on the Beyond Myrtle Rust website in the next couple of days, and also emailed to everyone who registered. Now, the webinar series will be taking a break in September, but it will be back Wednesday, October the 12th. We will hear from Roanne Sutherland at the Department of Conservation, previously at Scion, where she's given us a webinar from before, but now over at DOC. She will be telling us about a new Myrtle Rust monitoring and surveillance program DOC has been setting up on the conservation estate here in New Zealand. And it's going to be great hearing about preliminary results from that, as well as plans for that through the upcoming Myrtle Rust season. That's it for today, everybody. Haere rā, and see you next time.
Thanks so much.
Thank you.